It's my pleasure to introduce Dr. Anindya Dutta. As Dr. Nihal said, he is currently a professor, the Department of Genetics, professor and chair of Department of Genetics at University of Alabama, Birmingham. And you may not know that Dr. Anindya Dutta is an alumnus of CMC. After completing his MBBS, he was at IICB, started his basic science career there. He worked there for 1.5 years, and subsequently he moved to US to do his PhD at Rockefeller University, and subsequently doing his postdoc at Cold Spring Harbor University. And after that, he did his residency, MD, in anatomic pathology at Brigham and Women's Hospital, Boston. After that, he was a faculty at Harvard. And then he moved to be the chair of Department of Biochemistry and Molecular Genetics at University of Virginia. And if you look at his research, you will realize that he has made significant contributions in the field of genomic stability, DNA replication, and non-coded RNAs. He's an excellent speaker. I've heard him many times. And when I started my PhD, when I listened to his talk, and then when he came and talked to me, I remember I was very scared. And he asked me a lot of questions. And over to you, Doctor. That's all.
Excellent. Thank you very much for hosting me. Thank you, Nihal. As some of you know, I'm here in Vellore for an alumni reunion, but I decided to mix business with pleasure. And I've been a great fan of this stem cell center. I think it's one of the miracles of Vellore that it has grown. It's so successful, and there's been a perfect succession. I've always known of Nihal's work, and so I'm delighted that Nihal, you are the director now. I think you'll take it to great heights. It's wonderful to see so many people doing research at CMC. I'm going to talk to the students today afternoon, and I'll mention that CMC is amazing in its clinical work. It really is outstanding. I've never seen it's comparable to the best in the world, but I'm beginning to think that even in the research section, it's going to go much higher, and I can see the trends. So this is going to be an incredible journey that you are on. So it's a pleasure for me to be as part of this journey. So I decided to give a sort of more general talk, and it's really geared for the trainees and junior faculty. I want you to think that science is not a sort of a defined loop. Even though we write these grants as though we know what we are looking for, but what you have to watch out for is the things that you are not looking for. And then it's very hard to convince other people that what you have found is important or interesting. Don't give up hope. You have to sort of keep on nurturing that thought, building the experimental evidence, and see where it goes. So that's the story of tRFs. I'm going to introduce you to the microRNAs. This is the first family of short regulatory RNAs that have been highly recognized. And we and a few other groups discovered tRFs independently. And then, of course, the question became, these are short RNAs. The question is, are they junk? Are these just degradation products or something like that? Or do they do anything interesting? And that's the journey that I want to take you through. Again, there's no set path. Feel free to interrupt me. I'll repeat your question for those on the links, and we'll answer that, okay? So you heard of my journey through United States, and I wanted to tell you that the United States is a very large country, and I started off way out in the Northeast. I seem to have lost my pointer. Where's my pointer? Oh, here, it should be here. Good. It's out in the Northeast, and I've been progressively moving south through the years. And so I'm now in Alabama, where the weather is sort of like what Vellore is now here. But I like it. I love it because I grew up in this. One of the interesting things I discovered, it's a large country. And when I looked at the people in my lab, they seem to be becoming happier as we have been moving further south. And it's not because I've become older and wiser and nicer to my people, but it's something very simple: cost of living. So this is something that I like pointing out to people. There's gradients of cost of living, highly expensive California and down to the east, and then highly expensive Northeast and down to the south. So the result is, this is a way of introducing to the notion that the United States is still open for business. There are tons of places in the United States that you can go out and get training in and come back, whichever way you do. So don't get discouraged by all the chat that you hear in the background that, oh my gosh, everything is terrible and tough and grant funding is going down. It's all goes in waves, but it's a large country. There are lots of places to live in and lots of places to get trained in. I also like to use this opportunity to thank the many people who have come to my lab. All the people in red are from India. So it's a large list over the years. And you can see that a lot of them come back and do very well in this country. For example, you can see a guy who's at the Saha Institute, a guy who's at Terry Sass, JNU, and so on. This list goes on. And again, the purpose of this is to tell you that your name can appear on a similar list sometime. Just be ambitious and go for it. Don't be limited by what you perceive are your limitations. You can go anywhere, okay? And come back anywhere you want to. With that, the introduction to microRNAs. So you know this, microRNAs start out life as a long transcript called the primary microRNA, and it gets cut out by this RNase called DROTA DGCR8. The hairpin gets exported out into the cytoplasm. Another RNase cuts it called dicer, and then one of the strands gets associated with this Argonaut protein or AGO, and that is the effector complex. So the microRNA then recognizes messenger RNAs, brings that complex there, and ends up either degrading the RNA or suppressing protein translation. So that's the final textbook version of these. Of course, it didn't emerge like that. There's a very interesting story about it, and you can see all the people who have been highly recognized for their contributions to the story. Here's Andrew Fire and Craig Miller. They got it because they found an application of the pathway that microRNAs use, the short interfering RNAs. So you guys all know about that. But even when that award was being given, people are saying a short interfering RNA is something artificial. You throw in from outside the cell. Why does the cell have that mechanism? And of course, the people who characterize that are just got recognized last year, Victor Ambrose and Gary Rufkin, for microRNAs. So microRNAs are the naturally occurring short RNAs, and siRNAs sort of feed into that pathway. But it tells you the power of these discoveries. Whenever they were discovering these things, it wasn't obvious that what they're discovering was important or interesting. It's the subsequent work over decades that lends credence to that story. So with that in context, let me tell you about this tRNA-derived RNA fragments. So we are actually studying microRNAs. And what we discovered is that a completely new family of short RNAs that we call tRNAFragments. And the way we discovered is this. This was 2005 or 6, and we're starting sequencing microRNAs in tissues. In that case, the prostate cancer cell lines. So you get these short sequences back, and normally you map them against a database of microRNAs, and they say these are the microRNAs present. This is the frequency of different microRNAs, right? And this is where your eyesight and your brain makes a difference. The postdoc who was doing this, Young-Sun Lee, did the standard stuff, and he could have come to me with just those final results that these are the microRNAs expressed. But he comes back and tells me, but 2/3 of what you have cloned and sequenced doesn't belong to microRNAs. I said, why? How do you know that? He said, well, if I map them back to the genome, they map to parts of the genome that don't form hairpins. Remember in the microRNA biogenesis, it was a de rigueur that the microRNA forms a hairpin, so a dicer can cut it and release the short RNA. So these are not microRNAs. So what in the whole world are these? So then he goes back and maps them against the whole genome and comes back and says, half of these 2/3, that means about 1/3 of what we are sequencing, are coming from tRNA genes. You have to forgive me for saying, oh, I know what they're just degradation products. tRNA genes are highly abundant, and of course, the messenger RNAs are highly abundant. They get clipped, and these are the degradation products lying by. So that was the enigma. Are these really functional, or are these just degradation products, right? So we in fact call them something called tRNA-fragments, thinking that we don't know what their functions are. They probably are just fragments. But the first thing that Young-Sun Lee pointed out to me was that, so this is what our tRNA starts out as a precursor tRNA, a long transcript. it gets clipped by specific RNases to release the mature tRNA, and the CCA gets added at the end. So he said that if these are degradation products, they should be distributed fairly uniformly along the length of the precursor or mature tRNA. And all tRNAs should be producing approximately some of these fragments. But that's not what I am seeing. What I am seeing are ones that are strictly from the 5' end, about 22 to 26 nucleotides from the 5' end, and he called them tRF5s, or from the 3' end, and he called them tRF3s. And a subset were coming from this trailer sequence in the precursor, which get clipped out during the maturation process, and he called them tRF1s. They're very precise cuts. So degradation products, you could imagine, could be staggered, right? Cuts could be at various different places. But that's precisely single nucleotides that the cuts were happening to release these fragments. So we decided to take our life in our hands and just to publish this observation, and you can see that paper. And we still hadn't answered whether these could be degradation products. And this is the challenge that you're now facing. You're a student, you walk into my lab, and I say, hey, we have these short fragments. What experiments can we do to verify that these have any biological significance? So follow my line of thought and actually think for yourself first, what are the kinds of evidence you'll be looking for, and see if it matches up with what we looked for. And if you have anything unusual or that I didn't describe, tell me, because I love getting ideas that way, okay? How am I doing with the students? Everybody following me? Yeah? Actually, students, put up your hands so that I can recognize some faces. Hands up for whoever the PhD students. Okay, okay. All right, good. This is directed to you really, because what I'm trying to do is get you to understand how exciting science is. So you understand the problem, right? Here are the short RNAs. We think it's important. Nobody's going to give us a grant about it. So how do you find justification to study this? Is it worth studying? Might not be. So the first thing that I already mentioned were how precise these cuts were. You know, it really was just at this nucleotide or this nucleotide. If they're staggered, if they're at multiple different sites, and that could be degradation product. But the precision of the cuts was very surprising. So I told you the family members, the tRF5s, 22 or 26 nucleotides. This one is a little bit too long, about 28. tRF3s, 22 and 24 nucleotides, I think. And then there are these tRNA halves that were discovered in response to stress. And everywhere, one of the things that we noticed was no matter whose library of microRNA sequences you look at, they all had them. So this wasn't something unique to my lab in Virginia. It was just something that is being ignored because people just didn't know what to do with them, right? So everybody's library had these short RNAs and these very precise short RNAs. And the next thing that was peculiar was that suppose there's a given tRNA, most of the time, the most abundant tRF that you'd see would be just one species. If it was producing an abundant amount of tRF5s, it really didn't produce a tRF3. So there's some specificity both in the cleavage site and in the sequence that was being retained by the cell. So it gives you some hope that there's some biological significance. What's the next question one could do? Is how widespread it is. So this is a bioinformatician, Pankaj Kumar, who did this. He looked at libraries from all cell lines. One awesome power of bioinformatics, that people are now required to deposit their raw sequence libraries. So no matter what they say they have in the library, they haven't looked at everything. So we could pull out all those sequences and see how many of the libraries have tRFs. And you can see all these cell lines on the top row, right? All these tissues, all these organisms, including Cerevisiae and Pombi, which is very surprising, all these tissues have this tRF. Not only that, look at the y-axis, the reads per million. They're pretty abundant, tens of thousands per million. Most microRNAs are not that abundant. So that was encouraging. What would you do next to find out if these things are of any interest or not? So, what do you do with human beings when you meet an unknown person? We look at where they come from in the hope that we get some idea of who they are. And then we look for who are they hanging out with, who are their friends. So that way you get some idea about their character and what they might be doing. So we did exactly the same thing for tRFs. So we went back to microRNAs because they were the same size as microRNAs. And we first asked whether these enzymes, DROSHA or DICER, were important for generating the tRFs. And the next thing we asked is whether what company did they keep? Do they associate with this Argonaut protein or not? Again, we didn't have to do any experiments because people have been doing these experiments and depositing the sequence. People had published embryonic stem cells, which are wild type, WT up there, or DICER has been knocked out, the DICER gene, or DGCR8, which is an essential component of DROSHA, has been knocked out. So that those enzymes are gone. And when you looked at the tRFs, to our surprise, we saw that if anything, the numbers increased a little bit in the DICER and DROSHA knockout. So when he showed me this result, of course, the first thing I asked was, show me some positive control. Show me that something that is a product of DICER and DROSHA, does that really decrease? And the answer was yes. So these are conventional microRNAs. And if you compare the black bar, which is the wild type, and the colored bars, which are the ones that are mutated in DICER and DROSHA, what do you see? 100-fold decrease in the abundance of those microRNAs. So yes, the cell lines are exactly as advertised. DICER and DROSHA has been knocked out. MicroRNAs are gone, but the tRFs are still being produced. So didn't give us a great answer, said that the parentage of these things are not known. We don't know how the biogenesis happens. What are the company it keeps? Does it associate with Argonaut? So here again, we didn't have to do much. People had actually done this experiment where they cross-linked the protein of interest, which is in this case Argonaut, to RNA that has been labeled with thiouracil. Then they immunoprecipitate the protein, chip away all the RNA using RNases. So the only RNA that's left is what's protected by the protein, and then they sequence it. And basically, that's the way they identify what RNAs are associated with your protein of interest, in this case, Argonaut. Make sense? All right. What did we find? Well, as expected, these are various microRNAs, laid out on the x-axis. And these are the reads per million that are associated with Argonaut of some of them. And you can see that microRNAs, as advertised, associate very nicely with different Argonauts, right? They're up along this y-axis. What is surprising is that at least tRFs in 3s and 5s are extremely good at associating with the Argonaut. Argonaut 2 was missing in these experiments, but later on we find that associated with Argonaut 2 too. So we got a half and half answer. We don't know what enzymes are producing them, but we do know that these guys go and associate with Argonaut. What do you do next? You try to find out whether they do the same thing as microRNAs in the sense that they pull down a messenger RNA, a target messenger RNA, into the protein, into the Argonaut. And again, the experiment had been done for microRNAs. People have done this bar clip, and what they can show is the microRNA is associated with the cross-linked Argonaut protein, as is the messenger RNA. So you can even see the messenger RNA associated with the complex. And they find that the microRNA and the messenger RNA are complementary to each other over that first 8 nucleotides, the so-called seed sequence. When you looked at the tRFs that are associated with Argonauts, we saw exactly the same pattern. We found messenger RNAs that were also coming down with this immunoprecipitase that matched the tRF sequence. Not only that, the site of the cross-link is often marked by a mutation. Here there's a site of the cross-link, here there's a cross-link. So the uracil that gets cross-linked to the protein, when you do the reverse transcription, it acquires a mutation. So you can see the site of the mutation, and you can see if the anatomy of the tRF and its complementary messenger RNA are the same as microRNAs, and the answer was yes. So this was getting better. Even though they're not generated by DICER or DROSHA, it's associating with Argonaut, and it's coming in and associating with the messenger RNA by recognition of sequence of the messenger RNA. So we think. Okay? I'm going to take a pause just to see if I'm leaving the audience behind completely. Anybody has a burning question that they want to ask me? You're welcome to ask questions. Honestly, I encourage the questions more than my talking about it.
How about from the online sessions? Can they also?
Yeah, sure. You can ask from online.
So in your early slide, the expression of the microRNAs from human cell lines and mouse models. The expression in the human cell lines is, of course, is humongous, and in comparison to the mouse cell lines. And so, therefore, do you see the same kind of further experiments in the human cell lines also?
It's a good question, actually. After those initial statistics, we never went back. So what you really want to know is whether the tRF levels in the human cell lines relative to the microRNA levels, is that ratio remarkably different between what we see in the human cell lines versus the mice. Because remember, when you say reads per million, we are not saying how good was the harvesting of the microRNAs in those same samples, but we haven't done that. It's a very interesting point. We know that for tRF1s, the tRF1 levels are much higher in cell lines than in tissues. But for tRF5s and 3s, we don't know. Good question. Any question from the live audience? Sure, Shaji.
In the small RNA sequencing files that you analyzed and others analyzed, did they see that these tRFs have exactly the same as the processed microRNAs? Or they are around 30 nucleotides?
Right.
A little far.
Right. Excellent question. So in that fragmentation chart that I showed you, bulk of the tRFs that I'm focusing on are the same size range as the microRNAs. So the tRF3s, 5s, and 1s. The ones created by stress, the tRNA halves, are about 30 nucleotides long. Unfortunately, the field is no longer making the distinction. They're calling them all tRFs. I don't think the long ones, the ones that are 30 nucleotides, can enter into the Argonaut complex. The crystal structure of the microRNA with Argonaut suggests that it doesn't have that capacity.
Yeah, but still it is 30. That means that there is a process in place to cut it exactly 30.
Right. Now,
because when we did this experiment of differentiating CD34 positive hematopoietic progenitors to erythroid cells, the late stages, we got this 30 nucleotide tRFs coming, not in the early stage. So it's exactly cutting into 30.
Yeah, the site of cut is very precise, and we really don't know why. We think that there could be some sequence recognition, but no one has done that experiment properly, right? But it's absolutely true, the site. And this issue that you pointed out of the tRFs increasing with differentiation has been seen over and over again. And I think that perhaps it's saying that differentiation is like a stress response. You know, the cells stop proliferating and more cleavage happens. I'm sure you also noticed, if you're not, you can check it. If you look at the abundance of the parental tRNAs by, say, Northern, that doesn't change. But the cleavage and that fragment changes dramatically, changes with differentiation and with stress like aging or external stress like radiation or whatever. And we don't quite know why that's happening, right? Okay, let's go on. So, seeing is believing. This is still, pretending that this is happening because in the Argonaut, we are seeing the tRF and we are seeing the complementary messenger RNA. But can you prove that the two are actually annealed to each other in the same molecule, in the same Argonaut protein? For that, there's a really wonderful, here's a summary what I've told you till now, is that they're bound to the Argonauts, they form a structurally similar complex, and we find these messenger RNAs that are complementary to the seed sequence of the tRFs in the same immunoprecipitates. But the question we're now asking is, are they associated with each other the way we are predicting? And for that, this is the coolest experiment in the world. It's called CLASH. So what they do in this experiment is that they pull down the Argonaut with the microRNA, which is in red, and the target messenger RNA, which is in blue. Then they use RNases to trim all the unprotected RNA. So all you are left with are these two fragments together, right? You immunoprecipitate the Argonaut out, and then you do this trimming, and then you ligate these two. Now you do the reverse transcription. So the cDNA will now be a chimera, a red part, which will be the microRNA, and the blue part, which will be the target. Now think about this experiment. There's no way you'll get a microRNA messenger RNA chimera unless they were together in the Argonaut complex, right? It's perfect. So of course, we looked at that data set to see what fraction of those chimeras started with a microRNA and went to a messenger RNA, And were there any tRFs there, which did the same thing, a chimera that starts with a tRF and that goes to a messenger RNA, right? Just gorgeous. So these are the reads. Let me color them for you. The red part of all these reads are tRFs, one particular tRF. The blue parts is the same messenger RNA. Beautiful chimeras. And the point you'll notice is that the ligation points are very different. What that tells you is that this wasn't just one molecule where the tRF was associated with the target and it came down, and then by PCR we amplified the hell out of it, because in that case there'd be exactly the same chimeric site. It's multiple Argonaut molecules have the same tRF with the same messenger RNA annealed together. And indeed, when you stare at the sequence more, you find that the messenger RNA has this sequence that's in blue that's complementary to the tRF. So this was about as good as you could do by bioinformatics to say, we think these tRFs are behaving a little bit like microRNAs. They're associated with Argonaut, bringing down a protein with an RNA with it, and perhaps doing something to the RNA. The other thing that was surprising in this was that when you looked at the abundance of the chimeras, like what fraction of them, which is in red, start with a microRNA and goes to a messenger RNA, and in black is what fraction starts with a tRF3 and goes to a messenger RNA. And you don't need to look at the numbers. You can see that, to a surprise, the tRF messenger RNA, tRF-RefSeq gene chimera levels were like 2 to 3 times higher than microRNA messenger RNA chimeras. And I still haven't got my head around this, because we are thinking these are like another small family of short RNAs, and yet in the CLASH results, they seem to be dominating the microRNAs in their ability to pull down messenger RNAs into the Argonaut complex, right? All right, so what I've told you till now is you can see that they're bound to Argonaut proteins and bring the protein target messenger RNA that's complementary to the tRFs. Yeah. There's a question from Dr. Nihal Thomas.
So it's maybe a very naive question. Is all this process, in particular the chimera formation, is it an alternative to epigenesis? Is it also age-related?
None of those questions have been studied properly, and they should be, which is if you take early tissues and old tissues, is the number of tRFs with increasing, number one, is the association of tRFs with messenger RNAs, in other words, forming those chimeras in our experiments, is that increasing with age? None of that. And forget about age. If you look at Shaji's question, even doing differentiation, no one has looked at whether these things change. Like what we are essentially coming up to, what is the physiological variation that happens? And could this be relevant to the physiology we are studying, right? Yeah?
RNAFragments using the same kind of machinery for processing.
Yeah.
So do they have any competition between the microRNA and RNAFragments?
Absolutely.
That makes the difference.
Yeah. Let me repeat the question. He's saying if microRNAs and tRFs are using the same processing enzymes and proteins, is there competition between the two? So there's a little bit of an evidence in the Dicer knockout where we found that the tRF levels actually increased relative to when microRNAs went down. You notice that? So that made us think that that's a possibility, actually, exactly what you're saying, that when Dicer and Rocha are not there, then the dominant family of short RNAs become tRFs. However, I also told you that Dicer and Rocha are not being used to produce tRFs. So there shouldn't be a competition at that front. But another point that comes out from that is competition for Argonaut. Does that change? In other words, you know that there are cancers where Dicer is mutated and Rocha is mutated. So in those cancers, do tRFs become the prominent regulators? Has not been addressed. So it's an excellent question, but has not been addressed. Okay?
So the that they are associated with tRFs, whether the protein level of those mRNAs which they are going to synthesize are going down or anything is happening to those mRNAs.
Yeah. So you're essentially asking the next question my reviewers asked is that, well, this is all fine and dandy. You're showing me that tRFs are associating with messenger RNAs, but is there any regulatory law? Does it decrease the level of the messenger RNA or the target protein? So thank you for that. That will be the next part of this talk. So I'll show that to you in a second. Okay? All right, so with your permission, I'll go on. So this has now become a big field. So if you are interested, you can go to various databases that have tRFs. This is our database. Pankaj set this up called tRFDB. There are several others. You just have to do a Google search and you'll find them. And there are now over 2,000 published papers on these guys. So obviously there wouldn't be so many people working on them if the answer to your question was negative. In other words, if the tRFs did not regulate something. So I'm going to show you the evidence that's been building up to support the notion that tRFs are actually regulatory. Okay? So that's basically a question in red, which is experimentally, can we show that tRFs really regulate gene expression? So again, a few years went by and a really smart postdoc came to a lab called Janan Kustu. And what she did was this experiment. She transfected cells with a tRNA gene. And she showed that that increased the amount of the tRNA, not unexpected, but to our pleasure, that also increased the amount of the corresponding tRF3. So we now had a way where naturally tRF3s could be produced from a transfected tRNA. So the first question we asked, and those of you who have worked on microRNAs are very familiar with this, is if you take an artificial gene, a luciferase, and in its 3' UTR, you put in a complementary sequence to this tRF, right? Will this luciferase be repressed when you transfect in more and more of this tRNA? Make sense? Very straightforward experiment. Of course, there'll also be a control luciferase which doesn't have that target site. So it's not like global protein synthesis. You're going to look at whether your specific luciferase with the target site is repressed relative to the control luciferase, which does not have the target site. The results were really cool. So let's just take one color. So here it is in blue. In blue is a luciferase that has the complementary sequence for tRF3-3. I don't pronounce the 2 zeros for simplicity. tRF3-3. That's the complementary sequence. And here are the various tRNAs that are being transfected. The blue one produces the cognate tRF3-3. The red one produces 3-9. The green one produces 3-1. On the y-axis, you have the luciferase output. And what you can see straight away is that when the colors match, when the tRF that's being produced, 3-3, and the target luciferase has a 3-3 site, it gets repressed beautifully. But it's specific. The red and the green tRFs cannot repress the blue target, right? And you can see the same pattern holding true. So in one experiment, we could start addressing that the target genes are being decreased and that there's specificity, that's a sequence specificity. So it's similar experiments, I won't take you through all of them, made us test whether dicer was necessary for this process, and it was not, whether Argonaut was necessary for this process, and it was, and whether the seed sequence match was necessary. So we could make mutations in the target site and show that if you have mutations that prevent that matching between the tRF, then you no longer see this repression. Okay?
What about 3-5?
You mean 5 tRF5s. So tRF5s are much less functional than tRF3s. We can see them, but not for all of them, while tRF3s are extremely functional. So there's some distinction there that you haven't quite understood, right? And tRF1s, you almost never can do this experiment of repression. So I think that if they have any functions, they have different functions, right? Now, you might say, well, that's a luciferase reporter assay. It's an external gene whose regulation I'm looking at. What about internal genes, natural cellular genes? So, you can measure all the genes in the cells by RNA-seq. And then you can classify the genes based on whether they have a seed sequence match or not into ones that are targets. So the targets are in all these colored lines. In black are the ones that don't have the target sequence. And what you'll notice, and this is a CDF plot, I don't have time to describe to you exactly what it is, but what happens is if a certain class of messenger RNAs are being repressed when you transfect the tRF, the abundance of those messenger RNAs decreases, and this curve shifts to the left. So if the curve shifts to the left, the CDF plot, that says that the targets are being repressed by this transfected tRF. So what you're seeing from this is that endogenous genes also are being repressed by these tRFs. These experiments are, of course, first done with microRNAs to prove that this system works. So now we know that both endogenous genes and an exogenous reporter gene is being repressed by tRFs. So let's see what you have here. Yeah. We have shown you all of this. Well, unfortunately, I've given you what the next question obviously asks before people give you funds for this is, are these functional in a biological sense, say in a medical arena? Would this be useful? So the questions that you can ask is, are they prognostic in cancers if high level of expression or low level of expression predicts a bad cancer or a good cancer? And do they regulate cancer phenotypes, say invasion of cells or ability to form soft agar colonies or tumors, right? Any questions till now?
What are the mRNAs which were downregulated?
Right.
Based on that, we what would be the possible function?
Absolutely. If you do this experiment with microRNAs, you find so many different classes of messengers being regulated. It unfortunately doesn't tell you the primary function. So it's not a perfect system. It's many targets get decreased, and you can see certain pathways that are enriched. So for tRFs, for example, when you do that experiment that you're suggesting, what you find is neuronal differentiation specific genes are more enriched among the ones that are repressed. But we have never tested it in a neuronal differentiation system. And I'm looking around for a good model system lab that would be interested in looking at that. It's much easier to do it in model systems like C. elegans and Drosophila. But I wanted to stick to a cell line, so I looked at cancer phenotypes, right?
So if the endogenous controls themselves are being regulated, where the expression is really downregulated, so wouldn't that suggest that your tRFs are also sort of like, how good would that be if your endogenous controls are themselves downregulated?
It should be there, right?
Right. So since we don't know what enzymes generate the tRFs, surprisingly, there have been some candidates and almost all of them prove out not to be true, and other people go and repeat the experiment by knocking out the enzymes. We don't know exactly which genes would do the regulation. So we'll get there eventually when people are happy that a certain set of enzymes generate the tRFs, right? One thing that we do know is just like levels of any RNA is detected by the enzymes that generate, it's also determined by enzymes that degrade the RNA, correct? So not only can you regulate the level of water in a bathtub by turning on the faucet, or you can regulate it by blocking the sink, the output. So there are enzymes that are being identified that degrade some of the tRFs. For tRF1s, in fact, we recently published that XRN2 RNase is degraded, is important. So it would be worthwhile checking whether cancers, for example, where XRN2 is deregulated, does tRF1 change, but hasn't been done yet. For tRF3s and 5s, there's still a lot of confusion as to what generates them and also what degrades them. But you're absolutely right, that would be something worth checking.
Once again, one of the most rapid phases of growth and development is within the fetus and the placenta.
Yeah.
So I was just wondering whether the placenta would actually be a good model to study tRFs.
It would be a fantastic model to study tRFs. You are absolutely
abundance of tissue also.
Abundance of tissue availability, enzymatics. So for those of you who know in the microRNA field, when they found microRNAs to find the biogenesis enzymes, they made extracts from Drosophila larvae because they're very rich and they're easy to get. But Drosophila larvae are not as easy to get as placenta. So you're completely correct. In fact, if I were in a country like here where placentas are many, and it's easy to get, it's a discarded tissue, so it's not a problem actually. I would actually look at extracts and see if we can study the processing pathway. That would be a fantastic pathway to look at. Hasn't been done. Okay, so let's move on to these second parts. One question that we kept on coming is that, how do you know that these things are abundant enough? Maybe they're not as abundant as microRNAs. So fortunately, we could answer that quite easily by looking at RNA-seq of various tissues. In this case, we looked at lower-grade gliomas, for which people have done short-RNA-seq. And so you could look at that data, pull out the microRNA abundance, right? How many times a given microRNA was cloned, and the abundance of individual tRFs. And a very surprising thing emerged. So here on the x-axis is the number of microRNAs that have abundance of, say, 0, almost 0. Here, about 80% has abundance that's less than maybe 5 reads per million. You see that? The blue line is showing that. So that's the distribution of all these microRNAs that most of us study. They're very rare. We sort of forget it. Only about 20% are expressed at levels that we like. They're detectable, they could have some biological function. And among these, I've marked in red dots the ones that people believe have a role in glioma physiology. That's 7, MIR155, MIR124, and 15. And so you can see their abundance. So that's your positive control. So compared to that, how many tRFs have that level of expression? And you'll see with the X's, there are 3 of them that have expression levels at the level comparable to the microRNAs. So those we thought would be worth studying. So first thing that you do these days, thanks to TCGA, is look at the abundance of the tRFs in various tumors and see if they're prognostic. And that's what you show here. The blue line is lower grade gliomas that have a below the median level expression of tRF3-9. The red one is gliomas in the same cohort that have above the median level of expression of tRF3-9. And what you're seeing here is survival probability. And you can see that the patients whose tumors had higher expression of 3-9 did poorly. Interestingly, that turned out to be true for all 3 of them. All 3 of them are markers of poor prognosis. The higher expression seems to make a more aggressive tumor. So now we want to see some physiological experiment that supports this notion that this tRF can push the cancer cell into more aggressive behavior. So for that, first thing we tried is knocking down these tRFs. I mean, all these figures are saying the same thing, but let's look at these curves. What you're putting in as a locked nucleic acid that's antisense to the tRF. And as you increase the concentration of the locked nucleic acid for each of these antisense RNAs, you'll notice that the proliferation rate drops very nicely. So suggesting that each tRF seems to be important. And there are negative controls for this, of course. So relative to the negative control, the antisense to the tRF takes away the ability of the cells to proliferate, their viability. What about invasion? So we did the experiment there both ways. I'll show you the result one way, which is, you are familiar with this invasion assay. I'm sure people out here do them, where in the Boyden chamber, in the upper chamber, you put the cells, right? And then in the lower chamber, you put a very rich medium. So the cells have to migrate out through matrigel, which is what supports them, to the bottom surface of the membrane. So you're basically looking at the ability to invade and move through matrix tissue. And what you see is that if you take astrocytes, or if you take glioblastoma cells, and you overexpress the tRFs, you can see that the ability to invade increases. So this is consistent with the idea that high levels of these tRFs makes the tumor more aggressive. It makes them proliferate more, and it promotes invasion. Make sense? You're asking about what genes are regulated. So we started looking at exactly what you're saying, actually. We did RNA-seq, we found what genes were being regulated, and first we wanted to confirm that they're actually being regulated. And you can see that they're being regulated about the same extent as microRNAs. So let's look at SMAD1, a very well-known transcription factor. If you overexpress the tRF compared to the black bar, repression goes down. But if you put in a tRF that's got a mismatch, so it's not the same tRF, you no longer see this repression, right? And you can see the experiment the opposite way also. So here's SMAD1 again. If you put in a control antisense, this is the level of expression, but you put in an anti-tRF and the expression goes up. So it's looking real. It's looking like they do regulate genes and they do produce cancer phenotypes. We haven't yet arrived at the next step, which is to connect the gene regulation to the phenotype. So are there genes involved in EMT, for example, that are being regulated that will explain the invasion phenotype, or genes regulated to cell proliferation or apoptosis? And I suspect many people in the world are looking at that question right now.
What about other tumors?
So you see them in all tumors. I have intentionally focused on one type for funding reasons, but another tumor which is very rich in tRFs and where a lot can be done is hepatocellular carcinoma. For you guys, I've looked at leukemias, and you can find leukemias, and I've forgotten which one, AML, has got a very high level of several tRFs, but I haven't done any experiments with them. Definitely worth studying, right?
What about pancreatic? Very aggressive.
Yeah, I have definitely not looked at it, but it's a trivial experiment. Get your bioinformatician colleague to look at pancreatic adenocarcinoma and TCGA, and they'll tell you which tRFs are expressed and whether they stratify the patients for prognosis. We haven't done it. Right. Yeah.
So in the glioblastoma, there will be different stages, right? Progression, right? So these RNA fragments, it's like corroborating with the progression of the disease.
Yes, you're absolutely right. So you notice that we looked at the lower grade gliomas because there you could stratify between high and low. If you look at GBMs, which is of course the worst form, the levels are all mostly high. You're absolutely right. Yeah. Was there a question there?
In your bioinformatics analysis, did you come across a group of mRNA which can be targeted both by tRF as well as by micro?
Another excellent question. Basically, are there messenger RNAs that can be targeted both by tRFs and microRNAs? And of course there are. And my current grant has one sub-aim where I'm asking, do they compete, right? Or do they synergize? And we don't know the answer yet, mostly because no one in the lab has done that experiment yet, but we'll get there. Or somebody else will get there before us. But I think there's going to be crosstalk between the two systems. Okay? All right, I'm doing okay for time, yeah. So I'm not going to take you through all the presentation, but this is basically the same idea that we can take the 3' UTR of the gene that we think is the target, attach it to a luciferase, and we can show that that also gets repressed by the tRF. So everything is looking like, at least some tRFs have microRNA-like functions. Let's put it that way. Now, that's not the end of the story. Other people are doing other things. And so people have also found them in other contexts. So these people are saying that tRFs promote respiratory syncytial virus replication. These people, this was actually an obvious one. I'm sorry I missed it. I'm sure you guys know that retroviruses and lentiviruses like HIV use the 3' end of the tRNA to prime the reverse transcription. And these guys are smart. They said, the tRF3 comes from the 3' end of a tRNA. Does that help in HIV replication? And it does. It can actually prime the reverse transcription. There are functions that are completely unrelated to microRNAs that tRFs are beginning to find. So I told you that there's correlation with cell proliferation, though the mechanism is not known. This was a very interesting paper out of Mark Kay's group. Actually, this guy was a student in my lab and went to Mark Kay's group as a postdoc. And he found that just like microRNAs, there's a whole stream of stories coming out that microRNAs also stimulate protein synthesis. They don't always inhibit protein synthesis. They interact with the 5' UTR and turn up protein synthesis. He's finding the same thing on ribosomal proteins, that they turn on protein synthesis. And these are really beautiful papers out from some from Rockefeller and other places where they find that tRFs can titrate, in this case, a protein, YBX1, that is normally degrading oncogenic mRNAs, but because the tRF titrates it, it stabilizes the oncogene messenger RNA. This one is saying that tRFs have anti-apoptotic functions. That means they promote the survival of cells by titrating cytokine C. And then these are actually referring to the tRNA halves. These are experiments where they say that if you take, say, mice, and this is actually right up your alley, and you feed them high-fat diet, the gene expression pattern in the zygote changes. The question is, how does it change? And they ascribe it to tRFs produced in the father's epididymis, literally transferred to the sperm and carried into the zygote. It's pretty amazing. And there, those tRFs epigenetically repress the expression of specific genes in metabolic pathways. This, as you probably know, since you're an endocrinologist, it's been always around, right? Like nutritional status of the parents, how does it affect the child's future health? And this is one of the best examples of transgenerational inheritance. Another very fruitful area, which you guys can easily catch on in your favorite cancers, is to look at it in exosomes. So not only are tRFs in the cells, but they get kicked out of the cells in these lipid vesicles. And look at the number of papers and number of diseases. They've been looked at preeclampsia, normal prognosis, pregnancy, healthy donors, cholangiocarcinoma. Every single context, people have done this kind of experiments, and they find that they're present in exosomal vesicles. Not only the blood, this is the most amazing thing, people have found it in the urine. So again, what I'm telling you is that, when we study science, it feels like, oh, everything is done. In fact, my impression of many things is almost nothing is done, and nothing will be done. That's why I happily talk about it, because I'm hoping that somebody out here will be inspired to take it on, because it's a confluence of things. You need the systems. So in this case, you have this patient population that's incredibly diverse and rich. You need the institutional support, which you have in the form of this institute, where you can ask these questions. In your favorite disease, do the levels of the tRFs change in extrachromosomal vesicles, extracellular vesicles, exosomes? And indeed, people have done that. People are looking at these exosomal vessels, and they find that in different cancers, in other diseases, the levels change. So I think in the interest of time and to promote discussion, I'll stop here. And if anybody asks me a very pertinent question, I'll pick out the relevant slides of what's left in the talk. But what I want to leave you with is that endogenous tRFs are expressed at levels sufficient to repress gene expression. tRF levels are indeed altered in cancers. We have no idea how, because we don't know the biogenesis or degradation enzymes. And they're widely seen in extracellular vesicles, in tissues and fluids. And they've been proposed as biomarkers for various diseases. The field is absolutely wide open. All right, so let me skip this, and just in the interest of time, I'll just do the boring, excuse me. Let me acknowledge actually, since I'm not going to show you the results, I'm going to read out the summary. All short RNAs also get modified. In the whole microRNA field, there's a ton of literature on that. So in my lab, Zhangli Su started examining this, and she recently, not that recently now, reported that tRF3s have a methyl modification. On adenine position 1, a methyl gets added. The writer for that modification, the enzyme that adds that thing, is known. She discovered it. It's tRMT661A. In bladder cancers, she found that 661 levels are increased. So then the prediction would be the modification increases, and she found that. But the coolest part is that she found that when the modification of the tRF happens, the tRF is not that good at repressing the target messenger RNA. So now we are getting to a stage that not only do they regulate their targets, but the regulation process is regulated by enzymes that modify it. Doesn't that sound like epigenetics completely to you? Right? In epigenetics, the enzymes that modify the histones or the DNA, and they regulate the action of the nearby gene. In this case, there are enzymes like this tRMT661A that modifies the short RNA and regulates its ability to regulate its targets. So her prediction was that, oh, okay, this enzyme is increased in the bladder cancer, so that M1A methylation of the tRF will be increased in the bladder cancer, and it was. And her prediction was that the target genes that this tRF represses, in this case, a very fantastic set of genes came up, the unfolded protein response genes, they will get de-repressed. And this we know, actually. Cancers love de-repressing the UPR genes. It helps their cancer pathway. But instead of the tRF being altered in abundance, the modification is being altered, right? And now to thank the many, many people who have contributed this over the years. Young-Sun Lee started this ball rolling, and I have incredible admiration for that man. He's running a fantastic lab in Seoul. By the way, for trainees, consider also Korea, Japan, China now, and Taiwan and Singapore as potential places where you can get outstanding training. We sort of here, when you're growing up, you tend to think of the West as the only places, but there's really incredible science going on here. And in India, I just visited NCBS and University of Science, and the students told me stories of their discoveries, and they're comparable to the best in the West. So there are lots of good places now. So don't get frustrated by the choices that you have. Pankaj is a bioinformatician, has never touched a pipette man, and he made huge advances by helping me look through data that's been published by hundreds of labs around the lab. So almost the bulk of the beginning of the talk was his work. Janan, who's now at University of Tennessee, was the postdoc who did the experimental stuff. Manjari was another bioinformatician who worked in Pankaj and is back at the University of Hyderabad. This is Manjari here, in case you see her at a meeting. This is Pankaj. She's at the University of Virginia still. Zhang Li was the one who studied the modifications and is now an independent assistant professor at UAB. And Bree is an MD PhD student who is now a resident in pathology at Johns Hopkins. So these are people who really did the work. Thank you for your attention. I hope I've convinced you that it's a long process. The one thing about science I've learned is that it's much slower than I imagined it would be when I started. There are lots of false starts. You do something, you discover something, then no one believes you. You get very discouraged by bad reviews, but you have to keep on building the evidence. And eventually, if you're good at it, you do get a story out of it, and which is why I picked up this challenge. So from 2000 and when was it, 8 or 9 to now 16 years. And now we are all reasonably convinced that these are not junk degradation products. They're biological functions. And so the field has moved on to the next thing is what's the context, like what you're asking, physiological disease. What's the context in which these become important? Okay, so thank you for your attention. Questions? Have I heard from any of the students?
Sir, I have a question.
Yeah, sure. Are you a student?
No, sir, I'm a scientist.
Okay, hold on. I'm going to hold the students hostage and wait here until a student asks a question.
Sir.
Okay,
Sir,
you're a student, right?
Hello?
Hold on for 1 second.
Sir,
Okay.
Just a curiosity.
Hello?
In the initial experiments, you did a pull-down assay to check the tRF and the mRNA that's interacting. So I'm just thinking, if we have a RNA pull-down assay to check whether what are the proteins that are associated with this tRF, maybe it's like epigenetic proteins or kind of cancer-related proteins. Did you check anything like that?
So excellent question. And of course, we tried it and we're incredibly disappointed because we kept on finding boring RNA binding proteins. And even in the long non-coding RNA, there's much more realistic. These are short RNAs. So you could imagine that once you grab onto it, there's not much real estate left for a protein to remain bound to it. And the only proteins that remain bound are just high affinity RNA binding proteins. They're very boring to me, like splicing proteins and heterogeneous nuclear molecules. Another part of the lab studies long non-coding RNAs, where this experiment we thought would be more fruitful, where you pull down the RNA and then look by mass spec what proteins are associated. Bulk of what you find are common RNA binding proteins. And I cannot bring up myself to follow up. Suppose you're studying, let's say, a very fancy protein, P53, and you pull down P53, and the bulk of the proteins associated with this is actin and tubulin and very abundant proteins. You'll be very discouraged, right? You don't think that that's functionally important. And that's what happens when you do this experiment with tRFs. There might be better ways of doing it, but we haven't come up with that. But it's an excellent question. Yeah. But coming at it from the protein end is much easier.
Hello, am I audible?
Yes, you're audible and you can ask your question.
Thank you, sir, for a wonderful talk. I had one question, maybe a little bit of silly, but you mentioned in your clash experiment how you could ligate your target RNA and the mRNA. But in your results, you also found a consensus sequence which was complementary to the tRFs. So in your one of your latest slides, you also mentioned that you actually showed evidence that tRFs could actually promote translation. So is it possible to repurpose the clash experiment to actually find target of tRFs for different types of mRNA, or it's just for that ligation for only that specific experiment?
That's actually a brilliant suggestion. No, we haven't done that. So what's your name and who are you?
I'm a PhD student, sir.
Okay, what's your name?
Sir, Debanjan, sir.
Okay, good, Debanjan. That's a brilliant suggestion, and we haven't done that because you conflated two things, but you're absolutely right. You're saying that if you look at the chimeras, will you find chimeras that are the 3' UTR or chimeras at the 5' UTR? And perhaps when the tRF binds to the 5' UTR, perhaps it regulates protein synthesis in a different manner. That's really what you're asking.
Yes, sir.
We haven't done that. We haven't actually checked how many of our chimeras the target site is not in the 3' UTR. Yeah, it can definitely be done, and then we can check whether there's differential effect of the tRF on those targets. Has not been done. Good suggestion. Thank you. I hope I remember it till I get back to my lab. You can send me an email to remind me. Okay, any questions from, yeah, go ahead.
So I need some clarity. So when you did those bioinformatic analysis initially, so what percentage of these tRFs binds to the part of DNA that doesn't make sense?
Yeah, yeah. So in the first experiment, 2/3 of the tRF, of the short RNAs, do not map to microRNA genes. So things that form into hairpin. Half of those map to tRNAs. So about 1/3 were tRFs. 1/3 of the total population of short RNAs were tRFs. 1/3 were about microRNAs, sometimes about 40%. And then there's another significant percentage, if that's what you're thinking about 20 to 25% that don't map to microRNAs, don't map to tRNAs. So what are they? They map to ribosomal RNAs. So they're definitely a whole class of ribosomal RNA fragments, also very precise. And then they map to small nuclear RNAs, snRNAs. Not that I know of anybody working on those, but I think they're also worth looking at. Do they change under physiological or pathological circumstances?
A follow-up question. So has anyone tried changing the physiological conditions of the cultured cells and look at the tRF expressions? For example, simple physiological condition, if I keep the cells at high temperature, change the pH of the medium.
People have done those experiments, but I can't give you a summary of the results. I know that in my lab, we have done things like adding serum to make the cells proliferate or deplete serum to make them quiescent. And at that time, we are looking at tRF1s, and tRF1 levels definitely went up when the cells are proliferating, and not when they're made quiescent. But we haven't done it systematically to see whether individual tRFs change in different directions, for example. But what you're really pointing out is what I'm trying to say is that since it's a new field, there are a lot of easy experiments to do exactly like you're saying, right? Put cells in different conditions. You can walk into your neighbor's lab and look at differentiating cells, or cells put in quiescence, or cells mistreated with doxorubicin or something, and see if the tRF levels change. But
yeah, so what I'm trying to ask basically is it a systematic stress response that increases the tRF, or it's a random?
Yeah. So for the tRNA halves, it's definitely a stress response. It's a systematic stress response. For the small tRFs, tRF3s and tRF5s, we don't see that, right? So there we don't know what is regulating the abundance of these tRF3s and 5s. It's something that has to be worked on.
Okay, so one more question. So these tRFs are coming from tRNAs, basically.
Yeah.
And when you overexpress these tRNA genes, you see an overexpression or increase in the amount of tRFs.
Yeah.
So if you think of it as a systemic response, or it has a regulatory role, so when you see an overexpression of a tRNA gene, let's say some cancer cell line, it's the tRNA gene that is getting overexpressed, or it is the tRF that is.
Yeah, you're absolutely correct. And Zhangli is actually addressing that question. So it turns out, you know about RNA sequencing, where you get the whole messenger RNA-ome and you can look at small RNAs, and that's a small RNA-ome. It turns out that the tRNA-ome is not so easy. And the reason is that tRNAs have these nasty modifications that block the reverse transcriptase. So the reverse transcriptase doesn't go through. So multiple groups around the world, including some very famous labs, are working on mechanisms to remove the modifications and then do the reverse transcription so they get an accurate measure of the tRNA level and at the same time the tRF level, and then compare the two and see how often does the tRNA level go up, and that results in the tRF level, and how often are they completely separated and is the tRF crossing. I'll tell you in one circumstance where he checked it was in aging. In aging cells, we see an increase of specific tRNA halves in that case, not tRS, but tRNA halves. We don't see the corresponding increase in the tRNAs. So at least in that case, it's either the processing or the degradation of the tRNA halves is changing with aging. It's not the level of the parental tRNA. Okay, good questions all.
Okay, one last.
Can you hold on for 1 second? So if somebody else has a question, we'll give them a chance. Otherwise, we'll run out of time, right? So let's just hold on for 1 minute. Anybody else wants to take a question? Any of the
Can I ask
students?
a question online?
Sure. Are you a student?
I'm not a student.
Okay, I'll accept non-student questions.
Wonderful talk, Professor Dutta. Very lucid for a person who doesn't work in the area. Do these tRNAs actually serve as tumor suppressors in certain carcinogenesis? That means lower levels of them actually promoting an aggressive form of cancer. That's the first part of the question. And you very interestingly mentioned that these tRFs could be detectable in urine. I'm actually homing into cervical carcinoma, where today urine is used as a clinical specimen to very easily collect specimens from women so that you could prognosticate. Other prognostic markers are available. So these are my two questions.
Okay. So you're completely correct that there are people who are looking at urinary levels of all these short RNAs, including tRFs, to see if they predict outcome. I don't know of anybody who's doing it in the context of cervical cancer. The first obvious one that the people are looking at is bladder cancer and kidney cancer, because you can imagine that will be more immediate. But it's entirely possible that non-urinary tract cancers also could alter the levels of tRFs and microRNAs for that matter in exosomes. But I don't know of any current work on that. So that's your second question. And because I'm jet lagged, you have to repeat the first question.
My first question was, are tRFs associated with tumor suppression?
Yeah, we haven't looked at it systematically. There are tRFs whose levels go down in the cancer relative to the normal tissue. So there are people who have published those stories, but I haven't seen any functional studies where they have put in the tRFs and sort of decreased cell growth or cell invasion or anything like that, right? That's where it's headed. However, my reading is very sketchy, and a quick PubMed search might prove me wrong. So to my knowledge, I don't yet know of tRFs that are tumor suppressive, which is really what you're asking.
Yes.
They might be there, but I don't know of them.
Okay.
Good questions. All right, back to you.
Sir, I have one question.
Okay.
Two questions, basically. The first one is that
did you fall out? Okay, somebody fell out of the connection. Go ahead, you ask your question on site.
With the expression of tRFs, you also see a significant repression of the endogenous controls and
Correct.
in specific cell lines.
Yep.
And so could this be a feedback loop where the overall expression remains the same? So it is difficult to justify the expression of gene of interest, whether it is upregulated or downregulated, because it has a regulatory effect on the endogenous control first. So it's quite tricky to say that the gene of interest is upregulated or downregulated, be it cancers or under homeostasis. Second is, does this system operate in a systematic way where you look in a physiological situation. So if you say for HeLa cells or HEX cells or HT29 cells, where you have a specific number of cells, a characteristic type of cells, where you see these RFs being, upregulated, and then you see the downregulation of the endogenous controls. But could this be working together in a system where you have multiple different cell types expressing their own differential expression of these RFs?
Yeah, everything that you're saying is correct, which is that when you do this experiment, and this argument is as true for microRNAs or any regulatory process that you study, we always do the perturbation and we study the immediate change, right? But you're completely correct that there's entirely possible that there's a homeostatic loop back that then resets the regulation so that the level of the ultimate target is not changed. This argument is true for any regulatory process that we are talking about. But despite that, the equilibrium site changes in different circumstances. And what we're hoping is that we are looking at things that cause sustainable change in the behavior of the cell because the equilibrium level of the target messenger RNA is changed. No one has looked yet at feedback loops. This is true for microRNAs, by the way. This is true for transcription factors. Anything that you put into the cell, initially there's a perturbation, but if you let the system go on for a long time, many of them will settle back to the original level, but one or two might be at the altered level, and those are the ones that are responsible for the final phenotype. Similarly, in complex systems where there are multiple cells present, this is actually something that hasn't been done. As you know, there are now single cell approaches of looking at short RNAs and messenger RNAs. And to the best of my knowledge, I haven't yet seen anything where people have looked at how TRF levels are different in, say, different cell lines in a cancer, and if you see interaction between them across cells. So, perfectly valid questions. In the interest of time, I'm going to end with only one question from the audience because I put him on hold. So please go back to him. Identify yourself since you've asked some good questions.
My name is Anand Kumar.
And you're a what?
I'm a JRF.
You're a what?
I have a JRF.
You have a JRF. Okay, yes. Great. Anand, go ahead.
Yeah, so this is a basic question. Just wanted to know, why do cells upregulate tRNA genes?
Right. It's a basic question and probably one of the hardest ones, right? Why? So in physiology, people have seen that. Remember, the first problem is that till now, tRNA genes have mostly been studied by northern blots. So that means only a few genes have been looked at, and people usually don't look at the whole tRNome. Now that people are getting methods for doing the tRNome, we'll find other circumstances where different tRNA genes get upregulated or downregulated in different tissues. Why? It's a very, very problematic question, because if you increase one tRNA level and don't increase the others, it's not going to help the cell by increasing global protein synthesis. It will only increase the translation of those proteins where, say, that codon is enriched and that codon is rate limiting. But those are things that are worth exploring. And I think those people who are looking at tRNAomes, that's one of the things that they're hoping, that they want to find examples where an individual tRNA goes up and then protein synthesis of some proteins is changed. Of course, me being TRF biased, I'm tempted to think that an individual tRNA goes up to help produce more of that TRF. But I'm saying this out of complete ignorance. I don't know if that's true or not, right? So that's the why of it. We don't quite know. You really have to globally increase all tRNAs if you want to change global protein synthesis. But individual genes, they might change. Okay?
Thank you.
Yeah?
Maybe one more.
Okay, one last question.
Are there some dedicated RNAs for these tRFs? In other words, are the tRFs the only substrate for some xRNAs?
I don't know the answer to that. So you're basically saying, are there some messenger RNAs that don't have any microRNA target sites, but have TRF target sites, something like that? Any messenger
RNAs that can degrade these tRFs.
Oh, RNAs.
Yes.
You're talking about the proteins. So,
yes.
All right. So there's unlikely to be just a single TRF specific RNAs. But for example, XRN2 that we identified degrades TRF1, It also degrades messenger RNAs that have been clipped off from the, the cap. So it's unlikely that there's going to be a single RNase that's just dedicated to it. I mean, I'm of course talking out of ignorance because you haven't looked at all the degradation enzymes. The same is true for the RNases that generate them. Dicer and Drosha were kind of unique in that they look like, they're perfect for microRNAs, but they're not involved. Till now, no one has found a perfect RNase that cuts a cut and doesn't do anything else. So we'll see. That's waiting to be discovered. Nihal.
Thank you so much, Professor Dutta. I think what we have just experienced, we have heard of cutting edge research, but this is beyond cutting edge research. You could call this the Excalibur of research. And this is an Excalibur which has not been thrown into the water. It's still out there. And if you know what Excalibur is, then perhaps you need to read a little bit more on the story of Excalibur. But I think all of you need to hold on to the Excalibur of research. What he has also just shown us is that how various observations, accidents should always be taken extremely seriously. And I don't need to tell this to a group of researchers, but everything that you see is so important. And what we have thought of as tRNA and how it's generated with something else. The function is something totally different from what we ever imagined. And that is all about science. Let's give Professor Dutta a big hand for this extremely enlightening part. We wish you a wonderful time in India. This was the most cerebral talk I've ever heard in most recent times. But for your subcortical activities during the alumni meeting, have a great time. Thank you so much.
Thank you. Thank you very much.
Thank you once again, those of you who have attended over here and online as well. Thanks so much.
Sorry.